Start.
So start the class, sir.
Okay, thank you, sir. Good afternoon, everyone. This is Kumar from Tsrs&Jc Lingampally. I welcome you all for live online webinar classes. Dear students, yesterday we have been continuing the chapter genetics. Today we will continue the next lesson, next topics of the same lesson. Let's see what we have been learning. So from your second year zoology, we are learning sixth chapter, that is genetics, and this is the lecture 6. Okay, let's know what are the different concepts that we are going to cover from this chapter. In this chapter, in this class, mainly we are going to study about the two concepts, that is Human Genome Project, that is the concept, and second one is DNA fingerprinting. So under Human Genome Project, we will be learning about the aims of Human Genome Project, features of it, and advantages and applications of Human Genome Project. Under the DNA fingerprinting, we will be learning about different steps which are involved in the DNA fingerprinting and processing of taking the DNA fingerprinting. Then we learn about different applications of the DNA fingerprinting. So where do we use this technology? Okay, let's see. Right. Human Genome Project. First of all, we must know what is mean by the genome. Genome is a genetic structure of an individual. Okay, so one set of the chromosomes, one complete set of the chromosome can be called as a genome of one particular person. You see what is meant by Human Genome Project now. It was an international scientific research project, okay, with the goals of determining the base pairs that make up human DNA and of identifying and mapping all the genes of the human genome from both physical and functional standpoint. That means under this project, we come to know about all the genes of our human body. And there are 30,000 genes around in the body, and we will know about the sequence of the genes and function of particular genes. We also count how many genes are existing in each chromosome. Okay, so we have around 46 chromosomes, under which 23 are homologous chromosomes, isn't it? So we learn about each chromosome, and under that chromosome, what kind of genes are present, and each gene's base pair structurally and functionally. That means what that particular gene do, I mean what it does and how it is useful for us. There are different applications for Human Genome Project. So hope you have understood the basic meaning of what is the genome, Human Genome Project. It is to know about complete genetic map of the human beings. Let's see. Human Genome Project. Here in the slide, you can see a person and there are different chromosomes which are floating. So it is about to know about the chromosomes and genes which are present in the chromosomes. We already know that chromosome cell nucleus contains the chromosomes, and chromosome contain a DNA. Here in the slide you can see this is the cell, and cell has chromosomes, and when you see the ultrastructure of the chromosome, there will be a double helix structure, that is DNA, deoxyribose nucleic acid, and the part of the DNA can be called as a gene. Okay, so each gene produces certain proteins, and these proteins may be works as a hormone or the enzymes or anything else. Okay, So knowing about all the genes which are present in our chromosomes is the project, that is the Human Genome Project. The Human Genome Project was an international research effort to determine the sequence of the human genome and identify the genes that contains it. The US Human Genome Project is a 13-year-old effort which is coordinated by Department of Energy and National Institutes of Health, that is NIH. Okay. Let's see further about the Human Genome Project. Goals of the Human Genome Project. The Human Genome Project. Let's see what is this. What are the basic goals of genome projects? First is to identify all the genes that are present in the human DNA. That is the basic standpoint. Next, to develop genetic linkage map of human genome. Okay? And third, to obtain a physical map of human genome. That means knowing the actual base pairs which are present on the DNA. Next, to develop technology for the management of human genome information. So if we know which gene works for what, I mean, if you know the function of a particular gene, there is a chance of doing a therapy, which is called as gene therapy. We can supplement, we can make changes in the DNA so that we can avoid several genetic disorders. Let's see, to know the function of the genes, this is the most important part. And to determine the sequences of the 3 billion chemical base pairs which makes up the human DNA. Next, to store this information in public databases. Next, to develop tools for data analysis and to transfer related technologies to the private sectors. These are the basic aims, basic goals of the Human Genome Project. Now let's know what are the different features of the Human Genome Project. So after completion of the project, they have come up with some of the salient features. There are most important features of the human genome which are somehow unique. They are, see, human genome consists of the information of 24 chromosomes, 24 chromosomes, that is 22 autosomes and X chromosome, okay, and one Y chromosome. Look at here, why it is called only 24 chromosome. We have to know about it because we have 23 pairs of chromosome. Whenever chromosomes are arranged in pairs, suppose homologous chromosomes are 2 in number. So one chromosome will have the exact copy. That's why homologous chromosomes are similar to each other. So that is why we should not consider two pair of chromosomes. We should consider only one set of chromosomes in the body. So in that sense, out of 23 pairs of chromosomes, we only take 22 autosomes and one X and one Y chromosome. Okay, so around 24 chromosomes genome, 24 chromosomes can be called as a human genome. Why? Because other, whatever the rest of the chromosomes are there, they are just a copy of these 22 autosomes. Okay. So human genome means one set of chromosomes of the human body. Yesterday I have discussed about what is meant by homologous chromosomes and what is the complete karyotype of the human being. The human genome contains over 3 billion nucleotide pairs. Okay. Next, human genome is estimated to have 30,000 genes. Okay. There are 30,000 genes in our body and each gene has its own function. Average gene consists of, one gene consists of 3,000 base pairs. Now, what is meant by base pairs? Beside, there is a picture on the slide. You can see that nucleus, and in the nucleus we find the chromosome, and under the chromosome structure we find a gene, and gene has a ladder like structure, isn't it? So double helix with a ladder like structure, and each line which you are able to see on the DNA indicates one base pair. Okay, so in that sense each gene contains around 3000 base pairs. Okay, so it is called base pairs because they are made up of different nitrogenous bases. There are two types, purines and pyrimidines. Okay, so by nitrogenous bases it has been formed. But sizes of genes vary very greatly. Okay, and there is a largest known human gene, that is dystrophin. Okay, dystrophin is the largest human gene, and it contains around 2.5 million base pairs. Okay, around 25 lakhs of base pairs are present on the gene called dystrophin. Only about 3% of the genome, only about 3% of the genome encodes for amino acid sequences of polypeptides, and rest of all is the just the junk or the repetitive DNA. That means there is only 3% of DNA which is functional in our body, or whichever is discovered, and the rest of 97% of the DNA sequences are undiscovered. We do not know what is their function. So it is considered as a junk DNA. Okay, this junk DNA, if you observe the sequences of it, it has a repetition of the base pairs. They are called variable number tandem repeats, or it is also called as reflips. Okay, reflips. This is in normal English we call it as repetitive DNA. What is repeating here? The base pairs are repeating. Okay, the structure is again repeating. The functions are unknown over 50% of the genes. Only we came to know about 50% of the gene, and still we need to know about other 50% of the genes. Okay, let's see some of the other features. The repetitive sequences make up very large portion in the human genome. Repetitive sequences have no direct coding function, but they shed light on the chromosome structure, dynamics, and the evolution. So they are also useful. Repetitive sequences are also useful to know about, to do the research in the evolution process. Chromosomes, chromosome 1, the first pair of chromosome, and the first chromosome has most genes. Okay, more number of genes. That is around 2968 genes are present on the first chromosome, and Y chromosome is the shortest or the lowest number of genes are present in the Y chromosome. That is around 231. Almost all nucleotide bases are exactly the same in all people. Okay, but genome sequences of different individuals differ for less than 0.2% of the base pairs. So basic difference is seen in the repetitive pattern of the base pairs. Most of these differences occur in the form of a single base differences in the sequence. These single base differences are called single nucleotide polymorphs. Okay, single nucleotide polymorph. One single nucleotide polymorphs occurs at every 1000 base pairs of human genome. About 85% of all differences in human DNAs are due to SPN, due to the repetitive single nucleotides. Okay, so this is the basic difference between among all the individuals. Apart from that, all the human beings will have almost same genes, same base pairs. let's know what are the applications of the Human Genome Project. It means application of human genes and their functions. And to understanding of polygenic disorders such as cancer, hypertension, and diabetes. There are certain genetic disorders in the body, and to know about those disorders also, this Human Genome Project is useful. Next, to improvements in gene therapy. Alteration in the genes of the human to cure any particular disorder is called as gene therapy. Gene therapy is the most booming medical industry nowadays, and for research in the gene therapy, we must know about what are the functions of particular genes. If we know about the genes, then we can do some therapy to it. That's why improvements in the gene therapy are possible only by the Human Genome Projects. Next, improved diagnosis of different disorders, these different diseases. If we know the symptom of a disease, then we automatically come to know about the defect in the gene. Development of pharmacogenesis is also one of the applications of Human Genome Project. Hope you have understood. And now let's learn about another concept called DNA fingerprinting. What is meant by fingerprinting? Fingerprinting, why do we use it? So usually we talk about the fingerprints which are present on our thumbs or our fingers, isn't it? The fingerprints on the hands are usually be unique, isn't it? They are all unique. But in some cases, we do the DNA fingerprinting. Because DNA fingerprinting is highly accurate than the fingerprinting. So let's see what is the DNA fingerprinting, and in DNA fingerprinting, what kind of uses we can observe. First of all, let's know what is the DNA fingerprinting and how we take this printing. Satellite DNA regions are stretches of repetitive DNA. In our just now we have discussed in the Human Genome Project that we have around 97% of the DNA is only repetitive, isn't it? So this repetitive DNA is the basic point for the human DNA fingerprinting. So this repetitive DNA which do not code for any specific protein, okay, the phenomena is called restriction fragment length polymorphs. So these repetition of DNA is called restriction fragment length polymorphs. So it is also called as reflips. Okay, so RFPL, around 99.9% reflips are seen in our human genome. These reflips are called VNTRs. You must remember this word. VNTRs, the full form is variable number tandem repeats. These VNTRs acts as genetic markers, and these VNTRs are the basic components in the DNA fingerprinting. They are actually used components. These non-coding sequences form major chunk of the DNA profile in the human being. Repetitive non-coding VNTRs are unique to individuals. They are unique to the individuals. Since they are unique to individuals, we can identify, we can differentiate the person, and we can identify the actual victim of the actual person. What do? They depict a high level of polymorphism, and they are the basis of DNA fingerprinting. Any piece of DNA sample found at a crime scene, Okay. Suppose if there is a murder happen at one place, so they take different DNA samples, isn't it? So DNA is present in the cells, so they take out all the samples where they can find the cellular components. Like if they fight together, there will be hair which is fallen on the crime scene, and blood will also be there, because when they fight together, blood is a prime component which comes out, and saliva also contains certain cells, uh, and semen is semen or vaginal fluids also contain different cells which can give us a DNA fingerprint. So they take up the different samples from the crime scene, and they extract the coding of the DNA. So that's called DNA fingerprint of that one particular person. Let's see how does it happen. So, a DNA sample is taken from different cellular things, like from saliva they can extract, or some hair particles can also give us DNA, and vaginal or semen fluids. These structures, these components will have a DNA sample in it. They take the DNA sample and from them they extract the DNA. Then they do cleavage with the help of restriction endonucleases. Restriction endonucleases are also called as molecular seizures. What do? They cut the DNAs at their complementary sites. Okay, so in a complementary basis, they cut. So these DNA cuttings, the DNA base pairs, they undergo the gel electrophoresis to separate the DNA fragments. Here in the picture you can see. Next, the technique will be the southern blotting technique. They are blotted on a sheet. Here you can see using southern blotting technique, and then there will be the process called auto radiography to obtain the bands of the DNA. This is the main important step, and after obtaining the band structure, they develop and visualize DNA as a fingerprint. Here you can see the fingerprint of a person. We will see the examples of it as well in the next slide. Let's see, this is the suspect and rapist sperm and the victim sperm. So victims chromosome, what do? They isolate the DNA, then they cut the DNA with molecular scissors called restriction endonucleases. They separate DNA fragments by electrophoresis, and then they do the blotting techniques. Okay, southern blotting. Then they takes up the genetic map or the DNA fingerprint of the suspect and victim and the sperm. So then there will be the result. Let's see what happens in this section of victim's DNA. Here you can see there are 24 repeats of GT. GT are nothing but the nitrogen base pairs. There are 3 regions of repetitive DNA, and in the first region there are 24 repeats of GT, and the second region 16 repeats of ATGG, and 64 repeats of the ATT. Okay, so these are the repetitions which are found. Let's take this as a base, repetitions. And let's see the suspect's DNA. Suspect has 49 repeats of GT and 16 repeats of ATGG and 51 repeats of the ATT. So he is one of the suspects of the crime. And third is the crime scene hair. That means the murderer hairs. You can see there, their genetic map contain 49 repeats of the GTs, 16 repeats of the ATGG, and 51 repeats of the ATT, which says us, which is actually matching to the suspect one, isn't it? So victim is not a person, he is an innocent, but you can see the suspect's DNA, it is matching with the DNA in the crime scene. That means the place where the crime happened, the samples from where it is collected. So by this we can decide that the crime is committed by the suspect number 1. Okay, so the lengths of repetitive sequences match the lengths of the suspect's DNA. So the DNA found at the crime scene belongs to the suspect. This is how they actually find out the criminals in the DNA fingerprinting. It is more accurate, isn't it? Let's see an original DNA fingerprint, uh, which is taken under the investigation. Here you can see it is really hard to uh, know who is the uh, person who is committed, who is the uh, person who is committed, who is the criminal and who is not. But if we simplify this DNA fingerprint, it looks like this. See in the next slide, it looks like this. Here you can see this is the DNA fingerprint from the crime scene, and there are 3 suspects, 1, 2, and 3. Let us identify who is the criminal. See here, this is the 1 band, 2nd band, 3rd band, 4th band, 5th and 6th bands, and all these bands should be matched. So it is matching with the suspect number 2. See here. So fingerprint, crime scene fingerprint is completely matched to the suspect number 2. Hence, we can decide that the suspect number 2 is the actual criminal or the miscreant. Okay, let's see further. Now, let's say she is the mother and there is a child, and we must know who is the father for this child. Let's do this activity. So this is dad number 1, dad number 2, and dad number 3. Now we must identify whom this child belongs to, who is the daddy of this child. Let's see, child, whenever we talk about the child, it is the mixture of it is the a child will have both mother's and father's chromosomes. That's why we have to compare the child's DNA fingerprint with the mother and also the father. So here you can see child has this base pair which is taken by maybe by dad number 2 or the dad number 3. Okay, and next second band is taken from the mother. Okay, and here you can see the third one, it may be taken from the mother or from the dad number 1. So since he is not given any band to the child, dad number 1 is deleted because he is not the dad. It is fixed because in the first band itself we came to know that the dad is not this dad number 1 is not the father of the child. Next you can see this band may be obtained by dad number 1 or dad 3. Okay, and you can see the next band is also matching with the dad number 3. Okay, right, and the last band may be taken from the mother. So by this we can finally conclude that this child belongs to dad number 3 because all the genetic DNA fingerprint is matching with the child. Okay, this is how we can find out the actual father of the child. So it has this kind of application and it has many other applications also. Let's see what are the different applications of the DNA fingerprinting. Let's say it is useful in the crime scene. So if we take any DNAs from the uh, any samples from the crime scene, we can take out all those and we can take the complete blueprint of the suspect. Human relatedness, we can know whether the person is from which continent. We can also know about with the human remains and the fossil remains. We can also know the person belongs to which region, and uh, where he is related to, and uh, how better he is related to other animals. And paternity, of course, paternity can also be decided by human fingerprinting. Next, animal relatedness, of course, in the evolution. So in the evolution biology, we can correlate one animal with another by having this fingerprinting technology. We can know how related one animal to the other animal, and we can make the evolutionary links and the relationships as well. It is also useful for the anthropological study of the human evolution, and human apes and human beings is called anthropology. It is used in the anthropology as well. Next, we can know the disease causing microorganisms. Okay. Next, we can identify food as well. Next, human remains. Human remains can also be studied well by this. Next, monitoring the transplants, right? So by knowing the fingerprinting, we can know how best it is related to another animal, and transplanting can also be very easy, because and in the transplantation we must know whether the organ is related to the person, whether it can be fixed in another person or not. Otherwise, organs usually be rejected. This is called graft rejection. So during the transplantation also, we can know the relatedness to the one animal to the other animal. Okay, dear students, that is all for today. So the genetics chapter is complete. Hope you have understood. Thank you. Kashinath sir, the class is completed. Please end. Thank you, dear students, for listening to the class.